Huda Zogby is an MD from Baylor. She reports that in Rett Syndrome patients, there's a 20-25% smaller brain. They live to old age, and it's not neurodegenerative. MECP2 is a gene where the methylcytosine is followed by a proline, hence the name. And there's a spectrum of phenotypes seen in females with MECP2 mutations. There's classic Rett, there's mental retardation with seizures, Angelman-like, there's autism, and mild mental retardation and normal intelligence. It's a little big spectrum. It said 99% of Rett patients have sporadic mutation, whereas about half to 1% are familial, which I found interesting. There's a non-random inactivation of the X chromosome in Rett syndrome. And in the Rett brain or blood, 50% of the cells express maternal and 50% express paternal X chromosome. And so in the patients with autism or mild mental retardation, there's a smaller percentage of the maternal X chromosome being expressed. And it's very those who are asymptomatic have a very, very small number with maternal X chromosome being expressed. So to summarize there, women have two X chromosomes, and one of them has to be silenced. And this is kind of a dosage response with Rett syndrome, in that the smaller percentage you have of the maternal X chromosome that's being expressed, the reduced phenotype one bears out. That males die at 1 to 2 years of age with classic Rett mutation, unless they have what's called 47XXY, in which their mutant X might be silenced. And some males can live into adult life with mild mental retardation or seizures, etc. Again, if they have that very unusual genetic combination. And then she went into great detail about her MECP2 mice and talked at length about their phenotypes. Said that they're normal within the first 4 to 5 weeks, and at 6-7 weeks they become hypoactive, spastic, and have tremors at 8 weeks. And they found a specific mutation they termed 308. So MECP2 308 has very similar alleles that cause classical Rett syndrome, and they're very similar to the human phenotype. And they display an increased levels of kind of an anxiety-like behavior. They tried to rescue with wild-type MECP2, but overexpression, twofold overexpression of MECP2 in mice, causes a progressive neurological disorder and premature death. And she said that there's seizures induced by this overexpression that were verified by another researcher in Texas named Jeff Nobles. He's quite famous for his electrophysiology work. And so he said gene therapy is really not an option because of the dosing problems associated with MECP2. The phenotype of MECP2 duplication, so if there's two there, is that head growth decelerates, facial and axial hypotonia, and progressive motor delay, etc. So that MECP2 duplication is common submicroscopic cytogenic defect in the sporadic autism. Said that mice with the MECP2 duplication have a social defect, as judged by the time involved in greeting a new mouse in a barrier partition system. And in fact that there's high anxiety in the duplication mouse as well. And also mentioned that the 308 variant of the MECP2 has enhanced stress-induced corticosterone levels. And using chromatin immunoprecipitation, they found methylated cytosines in the promoter region. So this goes back to what some people believe as a basis for methylation problems being prevalent in autistic children. I mentioned that with regard to X chromosomal inactivation, it starts off at 50%, but the cells with the unhealthy X chromosome do not divide as well, and as the healthy X chromosome expressing cells, so the difference really skews over time, which I found to be interesting. I hadn't thought about that before. The last speaker was Mark Meller, an MD from Al Einstein College of Medicine in New York, and he spoke at length about many different topics. It was an extremely difficult lecture to follow, to be perfectly frank, and discussed how a lot of different genes are involved or are thought to be involved in autism. And he really talked up a lot of new techniques such as laser capture, microdissection, and real-time, or what is also known as qPCR, being able to show different gene expression patterns in different cells in, say, wild type versus autism models in mice, as an example. He spoke at length about medium spiny neurons and the different genes involved there, and trying to relate that to autism. And then he went on to show that a lot of other genes involved with glial cells, specifically oligo2 and MASH1, involved in both glial cells and stem cells, may be as well involved in autism. I have to say I wasn't entirely convinced because he was going so fast during this particular talk. But for me, probably the most interesting portion of his talk was him talking about the imprinted regions on chromosomes 15q and 7q. I thought those were really cool. He talked about these allele-specific genes and how they display preferential expression in the nervous system, and they're often transcribed from large genomic regions encompassing multiple tandemly repeated microRNAs and the CD small nucleolar RNAs, or SNO RNAs. He talked at length about chromosome 15. Again, this guy was going a mile a minute. And he mentioned his a paper, Trends in Neuroscience, 28, looks like issue 28, page 349, years 2006. And then as well tried to wrap up with some RNA editing and DNA recording. Said that 2% of the protein encoding genome is being actively edited, and mentioned that there might be a relationship between RNA editing and autism. Hi everyone, that's all I have, and what I really wanted to do is kind of show you what someone's notebook might look like after they sat down and listened to a bunch of very interesting but varied talks, scientific talks about autism, and give you a little taste of what that is exactly like. Anyway, thanks a lot. Bye-bye.